joining the last. We had a few just joining the last couple of minutes, so I think we're good to start. There's still a couple arriving. So anyway, it's a pleasure to introduce the next Advanced Plant Growth Centre seminar, and we're very fortunate today to have Matthias Benoit from INRA in Toulouse. He's worked previously in the UK, the Sainsbury Lab and the Cold Spring Harbor Lab in the USA. And his interests really are around sort of genome evolution and dynamics, and how the genotype shapes the phenotype in response to the environment. I'm looking forward to your talk today, Matthias. The floor's yours.
Rob, thank you so much for this kind introduction, and I would like to thank you and Robin for this very kind invitation. It's really a pleasure to be with you today, and I also see some familiar names in the attendees, so it's good to have everyone here. So let me just share my screen, and while I do that, I will tell you today about a story telling why gene duplications can actually be a barrier to engineer desirable traits in crops. So most of this work has been done during my postdoctoral studies done at Cold Spring Harbor Laboratory in the United States, and that's been finished in some extent when I joined INRA in Toulouse a couple of years back. So as you know, the modern agriculture really relies on a few numbers of major crops coming from three different main plant families. So you have the main crops coming from the grasses, the wheat, rice, and corn. For the legumes, we have soybean, and potato and tomato are two of the major crops coming from the nightshades group. So of course, the threat, the combined threat of climate change and an expanding worldwide population requires a way to strengthen and diversify our food production. One way to do that would be to include in our food crops production more diverse type of plants. One nice opportunity comes from the so-called indigenous crops. Indigenous crops are heterogeneous groups of plants that are largely related to the major crops, but that are bred at a much smaller scale in some specific location, and usually that have been undergoing much less important breeding and domestication. I'm showing you some examples here, such as the indigenous crops millet and teff coming from the grasses family, cowpea, pigeon pea for the legumes that are widely cultivated in Africa, and in the nightshades family, African eggplant and pepino are also indigenous crops that could represent an interesting investment to diversify our food production. The main issue of those indigenous crops, if we want to introduce them in our industrialized food systems, is that they are have been lacking really in depth domestication and breeding, and they still retain quite a lot of ancestral traits that are undesirable for modern agriculture. One way to fix that would be to use the knowledge we have acquired over the years in the modern crops, such as deep genetic knowledge in terms of DNA mutations and how this is linked to the phenotypic knowledge. Using genome sequencing and genome editing, that are technologies that have been immensely improving over the last few years, it's commonly assumed that we could transfer the knowledge from those major crops to those indigenous crops in a way to introduce beneficial mutations in those indigenous plant species to make them more suited for wide-scale food production, and also to remove those undesirable ancestral traits that could be limiting yield, by example. So that's a nice idea, but our work and others has shown that the introduction of those desirable mutations in new backgrounds is often blocked by what we call background dependencies. Background dependencies are specific genetic modifiers that will modify the effect of the introduced mutation, and that will hence cause unpredictable phenotypic outcomes. On the example you see on the right, you have here your reference species, let's say tomato, in which you have a desired mutation that will, by example, induce an increase in flowering time, which is a trait of interest in tomato. The idea will be by introducing these traits in the different rated species, such as other indigenous crops in the Solanum, will be to basically try to achieve the same phenotypic outcome using the same mutation. This could be done relatively easily using genome editing. But there are several examples where by doing so, we actually, at the end of the day, don't recover the phenotype of interest we want. There are many examples where, by example here, mutation in a flowering regulator does not translate in the same phenotypic output between those species compared to the major crop reference species. And this is due to the interaction between the de novo mutation inserted by genome editing and background polymorphism, genetic modifiers that are already present in the genomes of the receiving plants that will modify the activity of the mutation and will then modify the phenotype at the end of the day. So that's a big problem, of course, because if you cannot engineer your plant in a way that will obtain an expected phenotypic outcome, then this directed breeding becomes much more complicated. Our work in tomato has shown that gene duplication can be a major source of those background dependencies, background modifiers. So what are gene duplication? So due to genomic instability, a duplication of a gene or of a chromosomal segment can happen, ending up in generating two identical copies of the same gene, right? So those two identical copies, because they're exactly the same, they create what we call some genetic redundancy. Once the duplication is done, the fate of this duplication can be very different. The duplicated copy can be either totally lost, will be purged away from the genome, or in cases where the two copies of the gene will be kept, different scenarios have been observed. We either undergo what we call a gene dosage fate, where the two genes will be kept and they will basically be sharing the function conferred by the original gene. So there will be some redundancy that will be instated in the function of those genes. One will be able to compensate for the others. There could be some partitioning of the ancestral function of the originally duplicated gene, with the new copy taking in charge some specific function that were associated with the ancestral copy. Or we have some case of neofunctionalization, where there will be some innovation in the function of this duplicated copy, and this duplicated copy will basically gain new functions that were not present in the ancestral copy. So the fate of duplication can be extremely diverse, as you see. And what we've been showing using tomato as a model system is that those gene duplication, they do shape extensively several traits of agricultural interest in tomato, including flowering time and plant architecture, inflorescence branching and flower number that ultimately translate in improved yield, and also fruit size. So we have a good sense that those duplications can be an issue when we try to modify desirable traits in tomato. But for now, we still lack a good understanding if that's also true at larger evolutionary scales, and especially in the case where you would like to engineer phenotypes in species that are evolutionary related, but that evolved quite a long time ago from each other. So to do that, we decided to use Solanum as a genetic system. Solanum is one of the most species-rich plant genera. It's really ecologically diverse and also economically important. Why? Because it contains at least 3 major crops: potato, tomato, and eggplant. But also it registers quite an impressive number of the so-called indigenous crops I was mentioning in the introduction, right? So among the pepino and African eggplant that I already introduced you to, there's also minor crops such as naranjilla and others that are locally important and also offer a very nice alternative to diversify our food system. One other advantage of using Solanum is that there has been extensive genomic and genetic tools that have been developed in the major crops. We have good reference genomes for tomato, potato, and eggplant. And all the genome engineering toolbox are working wonderfully in this species. So this system, the Solanum, really allows us to test how gene duplication and paralogs, which are the term we use for genes originating from duplication, how they can first modify plant phenotypes and how they can actually be tweaked in order to really reach the desirable phenotype of interest. So to test this question, we decided to generate what we call the pan-genome of the genus Solanum. What is a pan-genome? So it's basically the collection of the entire set of genes present in a given species or in a given genus. To do this, we selected 22 Solanum species that capture the diversity of the ecology, the phenotypes, and the taxonomy present within the Solanum genus. You can see here on this map that we've been collecting species that originated from very diverse parts of the world, very different ecological origins, and with very different landscapes. So we grew those plants in the lab and we generated genome sequences using PacBio hi-fi sequencing, and we coupled that to long-range scaffolding data to obtain chromosome scale reference genomes for the 22 of those Solanum species. You can appreciate here at the bottom the diversity of the vegetative and fruit phenotypes coming from those Solanum. Those plants are really beautiful ones, and they really capture big diversity in terms of phenotypes. So once we have those 22 genomes, we looked at their diversity and we wanted to see how diverse and how much they evolve from each other. So those 22 Solanum genomes, they range from 700 megabase pair to more than 2.5 gigs in terms of size. So there is a real wide diversity in the genome size of the species. And what we've been detecting is widespread chromosomal rearrangements between the species. I'm showing you here on this plot, which is relatively complex, but the main message here is basically to show that you have here at the top chromosome organization from tomato. And each of the segment then, when you track down to the lower species here, basically exemplifies how much chromosomes have been rearranged between the different Solanum species. And as you can see, there's really been extremely broad rearrangement of chromosomes between those Solanum species. So among those 22 genomes, we could identify over 800,000 genes that were detected in all of the 22 species. And roughly 60% of those genes were shared with all the species we sequenced. So basically 60% of the genes were found in each of the 22 genomes of the Solanum we sequenced. We then dig deeper and we wanted to know how widespread duplication were in this pan-genome. So to do that, we used a tool called DupFinder that allowed us to identify close to 600,000 gene duplication found across the pan-genome, which is absolutely huge. And what we wanted to look at was, so to first identify the origins of those duplication. So gene duplication can emerge from different types of molecular mechanisms. The most common being whole genome duplication, as you can see here, where basically you are the entirety of the genome is being duplicated, so you end up with two different copies, two paralogs. But there are also different types of duplication, such as tandem and proximal, that really induce the duplication of a single gene that ends up creating two copies, and other more complex mechanisms that end up generating multiple copies of an initial gene. What is interesting, and it's plotted here on the right, is that most of those gene duplication, something again around 60%, most of those gene duplication are shared between all species found in the pan-genome, okay? So that's interesting, but not so interesting for our purpose, because if those duplications are shared between all species, it means that there is no variation, and basically they cannot make they cannot be genome dependencies then later on. What is more interesting for us is are the gene duplication that are not found in all of the Solanum genomes. So basically the light blue here and the green, those are gene duplication that are found in some, but not in all of the Solanum genomes, right? So generating polymorphisms between the different species and potentially then generating background dependencies. So now that we have identified our gene duplication within the pan-genome, we wanted to know how those paralogs, how those gene duplication would evolve in terms of transcription and activity, right? So here, let's consider a case where you have a duplication, as I shown in the introduction, right? So you end up having two genes that are identical in terms of sequence, in terms of regulation, right? So those genes will be here shown in blue and red, expressed at the same level and with the same expression pattern in all tissue tested, right? The question is then what happens when those genes start to evolve, right? What happens when those genes start to acquire maybe different patterns? So to test this, we had to generate a relatively exhaustive gene expression atlas of expression in Solanum, collecting over 270 samples from different type of tissues coming from the 22 species, right? So we basically tried to cover as much as possible the different transcriptome and specifically the tissue-specific transcriptomes coming from the Solanum species in order to understand how the paralogs would evolve in terms of gene expression. We computed that, and we were able to identify different paths of expression diversification between the paralogs. The first one was genes that were showing a pattern that we call dosage balance. This pattern basically is a large maintenance of this redundancy, where you do see conservation both in terms of expression level, but also in expression pattern between the different tissues, between the two paralogs, right? So you have the two paralogs, those would be expressed at the same level and in the same tissues, right? So those would basically generate redundancy and dosage balance. The second group we detected was the group that we call paralogue dominance. In the paralogue dominance group, you could see that while the two paralogous genes would keep the same expression pattern, meaning that it would be expressed in the same tissue, they do differ now in the expression level, right? With one paralog being more expressed, becoming more expressed than the other. Okay, so we basically have a shift in terms of expression, with one paralog becoming higher in expression and one staying lower. The third type we saw was a type of specialization in terms of gene expression. Here, the idea is basically very similar to the group 2 with the paralogue dominance, where you have one copy that decreases in expression in most of the tissue tested, except a few. Here, you can basically see that the red gene here, the paralogue number 2, is largely downregulated compared to the paralogue 1, but there are tissues in which this particular paralogue retains the same expression level and pattern, right? So that means that in this group, the new paralog could acquire some kind of specialization. It would become important and relevant only in some tissues. And the last group is group number 4, where we do see full divergence in terms of expression pattern and levels between the two paralogs. Here, as you can see, I think this one is self-explanatory. So of course, when you see such kind of pattern, it means that virtually all of them have the ability to complicate the genome engineering, right? And especially to make unpredictable the output of genome editing. Why is that? Because of course, if you edit one of the two copies, there will be the possibility for the second copy to either fully compensate the loss of the first one, or to compensate in some degrees the loss of the functional paralog, right? So of course, if we are not aware of the existence of such paralog in plant genomes, then it becomes difficult to really obtain the expected trait of interest at the end of the day after mutation. we wanted now to see how this evolution of paralogs would impact the phenotypes observed in the Solanum, right? And we wanted to know in particular how those paralogue diversification would influence major agricultural traits. I introduced them to you before, such as flowering time, inflorescence and branching flower number, so basically plant yield, and fruit size. So of course here we took advantage of the deep knowledge we have at the genetic level of the genes controlling those phenotypes in major crops, so such as tomato, potato, and eggplant. And from this list of genes, we looked at the presence of variation, presence of gene duplication and or mutation at those genes that are involved in controlling those domestication traits. So I'm showing you here a subset of what we found. Of course, we won't go in the details of the whole picture here. The main message here is that if you look at genes that were relevant for those domestication syndromes at the scale of the Solanum genus, we do find widespread paralogue duplication and or mutation in some of the genes, suggesting that those domestication phenotypes could be modified by the evolution of paralogs in those specific background. What you can see here is that really those, by example, taking the FTL1A as an example, you have really a broad variation in copy number and in the presence absence of mutation in those genes really across the entire genus, with the possibility of those mutations then impacting phenotypes at the end of the day. What I want to focus on here today is on this gene, which is called the clavata3. Clavata3 is a extremely important gene in the history of domestication of tomato because it's linked, so mutation in clavata3 is linked to an increase in fruit size, as you can see here schematized here that way. Clavata3 is a small signaling peptide that controls the stem cells at the level of the meristems and the flowers. So basically, when you lose clavata3, plants tend to develop meristems that over-proliferate because they over-generate stem cells. And they basically end up in what we call fasciated plants, so with stems that are much larger, showing some type of fusion. Enlarged flowers, and at the end of the day, enlarged fruits. And this increase in fruit size is largely driven by an increase in the number of seed compartments you have at the inside of the fruit, which are called locule number, right? So this clavata3 gene is a major domestication gene in tomato. And we wanted to know if clavata3 could also be involved in the evolution of traits across the Solanum, and if the paralogous diversification of clavata3 and the generation of gene duplication of clavata3 could also modify those phenotypes in Solanum. Oh yeah, so here I'm showing you basically the phenotypes I was mentioning before. So that's basically the clavata3 mutant in tomato, as you can see, right, compared to the wild type. The loss of clavata3 is generating those really hyper-developed enlarged flowers that end up producing larger fruits, which is really a trait of agricultural interest. So by zooming in on the important copy number variation and mutations, we detected that clavata3 in a subset of our pan-genomic genome. We basically found that there is a complex history of paralogue diversification at clavata3. So here you have a selection of the different species we targeted. And here you basically, I basically show the complexity of the haplotypes we detected in each of the species. So the clavata3 gene here is in green. And what you can see is that in some species, we detect single copies of clavata3, right? Such as in Cleistogamum, Torvum, Giganteum, by example. In some species, such as Prinophyllum, we detect a gene duplication of clavata3. So in this particular species, we do have 2 clavata3 paralogs derived from gene duplication. And we also have species that show that present pseudogenized or truncated version of the second clavata3 copy, right? So by example, Solanum ethiopicum here, Solanum anguivi, those show a second copy of clavata3 that is pseudogenized, so likely non-functional. So this diversity here is really remarkable because it shows how over relatively short evolutionary times, there can be really large-scale rearrangements at some loci, and how copy number variation in gene duplication can really generate extremely diverse haplotypes at the locus we're interested in. So we took advantage of this haplotypic diversity to test how gene duplication of clavata3 will modify the fruit phenotypes in those different species. So to do that, we selected 3 species. So we selected Solanum cleistogamum, which has a single clavata3 copy, Solanum Prinophyllum that has 2 intact and functional clavata3 copy, and Solanum ethiopicum that has 1 functional copy and 1 truncated copy. We took advantage of our CRISPR-Cas9 genome editing pipeline to basically induce mutations in the clavata3 ortholog in the 3 species we've selected, right? So you can see here the phenotypes of the loss of function mutation for clavata3 in the different species we mentioned. And what you can see here for cleistogamum and here for ethiopicum is that the loss of the 1 functional copy translates into this over-proliferative meristems phenotype that is very similar to the one we do see in tomato. And in Solanum Prinophyllum that has 2 functional clavata3 copy, you can see that knocking out the 2 copies by genome editing end up in developing a very similar over-proliferative meristem phenotype that is really characteristic of the clavata3 mutation in tomato. So the takeaway from this slide is that it seems that in terms of function, at least in the tested species, clavata3 retains the same function. But Solanum Prinophyllum gives us the opportunity to test something, in my opinion, even more interesting, and it's basically to test how redundant are the 2 clavata3 copies we detected in this species. So instead of mutating the 2 copies at the same time of 2 clavata3 copies of Solanum Prinophyllum at the same time, we decided to go for a strategy of targeting individual copy independently, right? Why do we see that there could be redundancy between the 2 copies of clavata3 in Prinophyllum? That's because in terms of sequence, those 2 copies are almost exactly similar. There's actually only 1 SNP in the 3' UTR of the second copy, suggesting that those paralogs originate from very recent duplication event. And in terms of gene expression, the 2 copies show almost perfectly balanced gene expression, right? So there is no dosage between the 2 copies. It really seems that the 2 copies are expressed at the same level. So based on that, our prediction would be that if we inactivate one of the 2 copies, the second copy should be able to rescue the phenotype and should be able to compensate for the loss of the second copy. So we tested this by inactivating 1 copy or the other. And here's the results, right? So here we're expressing the results in terms of the number of locules, right? So which is a proxy for the fruit size. And what you see is that Prinophyllum in wild type largely has 2 locules fruit. But when we mutate by CRISPR-Cas9 single clavata3 copies, we do see a bump, we do see an increase in the number of trilocular fruit, okay? Of course, when you mutate the two copies, you end up with fruits that are, well, multilocular, or let's say like, crazy fruits that really show totally aberrant structures and fruit development. The takeaway from these results suggests that even if the two copies seem almost identical to each other in terms of sequence and expression, they cannot fully compensate to one another. And that's because we do see an increase in plurilocular fruit between those two mutations, right? So that was fairly surprising to us, but it means that there is already some type of divergence between those two clavata3 copies that is happening in Prenophylum. But still, if you mutate one copy or the other, there's still large compensation of the fruit phenotype in this condition, right? So imagine a system where you would like to inactivate clavata3 in an indigenous crop where you don't know if there is one or two clavata3 copy. You may end up in a situation where you will actually mutate only one copy out of the two, and the second copy would still compensate for the other, right? So you will end up not having the expected phenotypic trait at the end of the day in your engineered plant, right? So that's one example of how gene duplication could actually be a barrier to predictable genome engineering and phenotypic engineering in plants. For the next few minutes, I would like to talk about a second aspect of our work that is still related, but that goes a bit even further in this notion of gene duplication evolution. And I will talk about African eggplant. So African eggplant, as I presented in the introduction, is a really major indigenous crop that is widely cultivated in sub-Saharan Africa and also in Brazil, where it has been brought by enslaved Africans. And Solanum ethiopicum, African eggplant, can be agriculturally grown either for its fruit, where you can see where you can appreciate the really wide diversity in phenotypes here, but also for its leaves, because you can consume African eggplant leaves, and there are also some specific accessions that are used as ornamental ones, right? So African eggplant really embodies, if you want, in some ways, all the potential that indigenous crops can have in the context of agricultural diversification. Of course, we were really stunned by such a big diversity in terms of fruit phenotypes and size, right? And the question we asked, we wanted to know if this diversification in fruit size could be linked, similar to what is seen in tomato, to a diversification in the clavata3 signaling pathway, in particularly a dynamic evolution of the clavata3 paralogs in this species. So to do that, of course, you need to capture the genomic diversity of the species. So we grew close to 100 accession of African eggplant in field conditions in New York. And we selected 10 representative accessions that basically cover the different cultivar groups. So Gylo, Shum, and Aculeatum. So those are the different subtypes of the species. Alongside the wide progenitor of African eggplant that is Solanum nguevi, right? So we generated reference genomes for those accessions using PacBio Hi-Fi, and we generated annotation for those species in order to have an idea of how diverse those genomes would be and how widespread gene duplication could be in this species. So it wasn't so surprising. We detected, of course, a large number of structural variants between those accessions, close to 250,000, which is surprisingly similar to what we detected in our pan genome of tomato. So it really seems that the two species show very similar genomic diversity. Of course, because we're interested in fruit size and fruit phenotypes, we wanted to know if some of the structural variation was related to clavata3, which, as you know, is a major regulator of fruit size in tomato. And what I'm showing you here is basically the presence, absence, and copy number variation of clavata3 in all those species. While most of the accessions of African eggplant do have the typical 2 copies duplication of clavata3 with one being non-functional, as I showed before, there is one accession showing an interesting pattern with seemingly having only one functional copy. So we dive into that and looked into more details at what happened in terms of genome structure at this locus, and microcentennial analysis actually showed very broad rearrangement at the clavata3 locus. So here I'm showing you centenic region between Nguivi, which is the wild progenitor, the reference Ethiopium genome, so the one with the one functional and one non-functional clavata3 copy. And here you can appreciate how widely different the clavata3, what we call deletion accession, is compared to two others, where clearly we could actually detect a very large DNA deletion, a very large genomic deletion, close to 300 kilobases deletion between the two clavata3 copies. And this big deletion, instead of fully removing one of the two copies, it actually created a new functional copy that is derived from the fusion of the two ancestral ones, right? So imagine that you have the clavata3A copy, the clavata3B copy. This deletion that encompasses the two of them ended bringing them together in a single fused copy that is functional. All right, so that was a very surprising genomic finding, but it really seems that in this accession, there is one functional clavata3 copy that is derived from the fusion of the two ancestral ones. So here, basically have the reverse of a gene duplication. You have those two genes that end up being merged in a single functional one at the end of the day. We wanted to know what would be the impact of clavata3 on the fruit phenotype in Ethiopium. So to do that, we decided to use a QTL6 strategy, where basically by crossing Ethiopium accessions with low local number with Ethiopium accession with medium local number, we could then map in intraspecific Ep2 population the genomic regions that are associated with the phenotype of larger fruits, right? In this specific population, so low local cross to medium local, we were actually surprised not to detect any clavata3 signaling members or any clavata3 genes linked to this phenotype. What we actually detected is a major effect QTL that contains a gene that is a serine carboxypeptidase encoding gene that has important function in the processing of the clavata3 peptide, right? So in the medium local number accession in the gylo, there is actually a small deletion in the coding sequence of the gene that generates an early stop codon, ending up producing a truncated protein. And this loss of function of the serine carboxypeptidase actually increase the fruit size in the medial local number accession by a few locule. So here we basically detected a new fruit size regulator in African eggplant that hasn't been found before in any other Solanum species, and particularly not in the major crops such as tomato. So we wanted to know what would happen if we would mutate this gene in other Solanum species, including tomato. right? So using genome editing, we mutated the ortholog of this SCPL25 gene in tomato and in Solanum prenophylum. And what you can see is that the loss of function of this gene in those two species end up increasing the number of locules in the fruit, right? And hence increasing the size of the fruit in those two species, right? So that's really an interesting finding because it suggests that there's mutations, so there are genes that have been linked to domestication phenotypes in indigenous crops that could be translated into major crops. So there's still a lot of knowledge that could be used from the indigenous crops and to brought it in the major crops. And we also did a second QTL-seq analysis, this time between so the same medium fruit size accession with the high fruit size accession to identify potentially other modifiers that would be involved in this increase in fruit size. And here we did see a totally different pattern with two minor effect loci that we detected in the high locule number accession. One of them is on chromosome 5, and we haven't been able yet to identify which gene could be involved. But for the chromosome 10 QTL, the peak just fall on the chromosomal location on clavata 3, and particularly the clavata 3 deletion allele I just introduced you to a few slides before, right? So in this particular condition, there is a minor effect phenotype that has been conferred by mutation in the clavata 3 ortholog in Solanum aethiopicum. So to sum up what I just presented to you, paralogue evolution and diversification has a major role in the evolution of domestication phenotypes between different species. In the case of Solanum aethiopicum, what I showed you is that the main driver of increase in fruit size is not clavata 3, it's this carboxypeptidase gene that was then fine-tuned by the addition of minor effect haplotypes such as the clavata 3 deletion. This is widely different from tomato, where in tomato, modification of clavata 3 was really the main driver in the increase in fruit size compared to the wide relative. And in this case, the carboxypeptidase gene hasn't been involved at all in this domestication process, right? So I think the important here is to know that across evolution and domestication, sometimes similar genes or gene families, especially paralogs, will be involved, but they may have very drastically different effect or be mobilized at different time in the domestication process. And then they will be also interacting differently with other genetic modifications that are already present in the genome of the receiving species. So with this, I would like to conclude with a few take-home messages. So I hope I convinced you in this presentation that gene duplications can really complicate trait predictability, and especially the fact that transferring traits across related crops can be unpredictable because of this hidden parallel variation. We can tackle that by leveraging pan-genomes that will really reveal the genetic diversity within and across species, and specifically coupling this pan-genomic and expression data will help to expose the evolution of parallel behavior across species. And that specific example of clavata 3 and the clavata 3 paralogs that really underpins fruit domestication traits. And the complex evolution of those paralogs have driven fruit size variation and domestication trajectories differently in different but related crop species such as tomato and African eggplant. So this work has been mostly done during my postdoc at Cold Spring Harbor in the lab of Zach LIPME. So I really want to thank Zach for the time and energy and support he gave me during this project. That was really a major collaborative work. I would like to really thank Mike Schatz and Kati Jinicki that were instrumental to this study, and also all the other collaborators involved. And I would also like to acknowledge funding for my current lab in Toulouse, that it was also very important in helping us wrapping up this story. And with this, I would like to thank you a lot for your attention, and I would be happy to take questions.
Okay, thank you, Matthias, for a very interesting talk. And no doubt you have a huge pile of data there, which I guess will keep you going for many years to come.
Yes, indeed.
So if you'd like to stop sharing your screen, that's great. So, Ramin, am I right in saying that we can't unmute people? Is that correct?
If they raise their hands, we can unmute them.
Yeah. So if anyone's got any questions that they'd like to ask Matthias, then please raise your hand, and we can unmute you and allow you to ask that question. If you prefer, I can answer that question for you if you'd like to type it into the chat. Okay. So Ramin, you've asked the question, yeah, which is, well, why don't you go ahead and ask it?
Wondering if there has been any studies on the flavor of it, with the size being bigger, or on the nutritional value of tomato.
Yeah. I mean, of course, we expect that this duplication behavior would impact virtually any phenotype, right? Desirable phenotype in those crops. So we haven't been looking at that, and I think it's not really in the follow-up of this study. So something that is important is that all the resources and the materials and the genome that I presented here are freely available as well, right? So if someone is interested into investigating this type of questions, the genomes are there, the expression data is there. So of course, one could really ask and look for the favorite genes involved in nutrition or taste, for example, in those species, and to see what's happening there.
Okay, so there's a couple of questions I'd like to ask, actually, Matthias. So in your initial analysis of the Solanum clade, you show that there are about 60% of your core gene. There's a 60% set of core genes which are duplicated across the entire clade. So does that mean then that duplication occurred in an ancestral species, or is it convergent evolution?
Yeah, so particularly the core genes, we do consider all genes, so not only duplicated ones, right? So this basically all genes. But it's true that we do find quite a lot of duplications that are found across all species, right? So it means that the duplication happened in even further ancestor of those species. So of course, here it would be interesting to complement with other studies that have been looking at the conservation of duplication across larger evolutionary times, right? You could imagine going all the way to grasses, by example, and to check if the duplication are there already or not. And then it will help pinpointing when that happened. So roughly, I think we can say that 40% of the duplication we do see in the pan-genome are Solanum specific.
And are there specific regions of the genome where you see a greater frequency of duplication, or does it tend to occur across the genome as a whole?
Yeah, that's a great question, right? I think so. Did we see hotspots of duplication? There are regions that are more prone to structural variation in general, right? So we know that regions with high transposable elements / repeat content, by example, those tend to generate a higher rate of variation and gene duplication. In the case of the clavata3 haplotypes I showed, by example, the clavata3 is really nested into really a bunch of transposable elements and if I remember correctly, NLRs as well, right? So highly repetitive gene families. So those regions are, because of their genetic structure, more prone to producing those duplication. Yeah.
And so another question I have was you. Oh, and I'll let Martin ask a question as he's raised his hand. If I just, I'll just unmute you, Martin, and then. Okay, sorry.
Hey, I hope you can hear me.
Yeah, we can.
Hi, Matthias. Nice to see you.
Hey, Martin. Nice to see you.
I was just wondering when you look at these diversification events for, let's say, certain genes like clavata3, how often does it happen that it's not the coding sequence that it's involved, but it's the UTRs or even upstream elements like promoters, enhancers, etc. Has anybody looked at that specifically?
Yeah, so that's a great question. We looked at that a bit, right? Because what you expect is that you would have a duplication most of the time, of course, of the coding sequence, but also the regulatory regions, right? That's something I haven't presented here, but we do also see specific dynamics of evolution of the promoter region, by example, right? Depending on the type of duplication you have, depending on in which group those duplication, expression group those duplication fall in, you will also have different patterns of evolution of the regulatory sequences, right? By example, you would expect that duplication that diverge very quickly in terms of expression pattern, by example, there would be more difference accumulating in the regulatory regions. So if you're interested, we can talk more about that in details if you want, so feel free to reach out. But it's also fairly well described in the paper, so you can have a look.
Brilliant. Thanks, Matthias.
Pleasure.
Okay, so I have another question. You also showed a slide where we're looking at paralogue diversification, and in some of those species, you had no copies of particular genes. So are those functions in those genes then picked up by genes that are duplicated in that same family elsewhere? Again, are there structural?
Yeah, that's a great question, and I'm not sure I have a good answer to that. I think eventually something you have to test is also the function of the duplication, right? So here we base our analysis on the type of duplication and the expression pattern. We also know that the evolution in the coding sequence, it has been shown for clavata3 by example, right? Evolution in the coding sequence might favor this type of subfunctionalization, neofunctionalization, and whatever happens when you have gain or losses of gene copies. That's all I can say really about it. I think, of course, it's worth investigating this type of question as well, and how basically the entire loss of those genes could be compensated elsewhere.
Thank you. Sanjeev has a question. Sanjeev, would you like me to unmute you and you can ask yourself?
Okay, yeah, thanks for a great talk. Yeah, this is.
Thank you.
I just want to know how much of the inferences you mentioned about duplication, they are affected by the bias in the pan-genome?
Yeah, that's a good question. I mean, I think how are we sure that we capture the entire diversity of the species or the genus in the pan-genome? I mean, of course not. We try to maximize the diversity we capture using those 22 species. We do capture most of the diversity really, but there are, of course, specific patterns or events of duplication, haplotypes we will miss, right? So I can confidently say that we do not miss the most obvious trends and patterns because of our sampling diversity. But there are more specific variation that could have been missed in this context, of course, because there are quite a lot of private duplication events, private structural variants that could also modify the outcome of what we're looking at here. So 100%, I think, more genomes in some ways is always better, and especially if you want to catch those really rare cases of variation.
So the pan-genome you mentioned here is the assembly of single separate reference genome. It's not like a true bubble kind of pan-genome, which is merged into one.
No, that's correct. Yeah, no, we haven't integrated all the genomes together. But that's something, of course, that would be interesting to do in the future, right? To how to integrate all those genomes, then to be able to assess in one go all the variation available there.
Okay, thank you. Yeah.
Thanks, appreciate it.
Thank you, Sanjeev. We next have a question from Susan M. I think, Ramin, could you unmute Susan, please?
Hi, well, thanks, Matthias. Can you hear me?
Sure, I can. Yes. Hi, Susan.
Oh, wonderful. Well, great talk and great work. I'm curious, have you looked at expression patterns and how perhaps they vary across duplications that are next to each other, proximal, tandem, or just proximal versus the ones that are far apart? I'm just thinking about transcription factor sort of occupancy and the competition simply because they're coming to the same location versus the ones that are more, I guess you could say, independently competing. And
Yeah, that's a
whether you
great
see those effects in expression. Thanks.
Sure. Yeah, no, thank you for the question. I think it's a great one. So we looked at that a little bit. What I can say is that duplications that are derived from whole genome duplications instead of local duplication, those tend to be more widely expressed. So they have a higher diversity in terms of expression, like tissue expression, by example, right? So they are more widespread, more broad in terms of expression, while the tandem ones tend to be more tissue specific. Would that be linked to the variability to transcription factors, like local competition? I mean, I don't know. I think it would be related maybe more to like of a dosage effect or like a large scale dosage effect where because of the whole genome duplication, you would have an entire signaling pathway that would be duplicated, right? So you need to really fine tune to make sure that the entire system keeps going. While when you have tandem duplication, it's only one part of the system, right? So maybe the difference in expression could also be due to this type of genetic structure. But the idea of competition, local competition, is also a good one. And I don't know if tandem duplication can affect the competition for TFs, by example. I think it's a good question.
Thanks.
Okay, thank you. Dave Cook has a question, which we'll just unmute and let you ask, David.
Okay, thanks, Benoit. Yeah, speaking as a plant pathologist working on potato late blight, I'm very interested in the NBS LRR genes for resistance, and presumably this is a big group of paralogs. I'm just wondering if you've looked at this group or have any plans to. Yeah,
great question. Of course, yeah, you're right. I mean, those genes are evolving so quickly and they generate so easily paralogs that when you look at the function of the duplication, of course, one of the most enriched function is of course those resistant genes. But we are not planning on looking in more details in that direction, right? So I think that would be fantastic to look into these kind of questions and again to see how at the sequence level, but also the expression level, those paralogs could evolve and then mediate resistance towards pathogen and others. So, if someone wants to do that,
scope for collaboration, maybe.
Yeah, 100%. I think, yeah, absolutely. And again, the resources are here, right? So if you can take advantage of it, would be awesome.
Fantastic. Thank you.
Thank you.
Okay, so I think we'll just take, it's almost 2 o'clock. We'll take one last question from Luke, please.
Hi, yeah, again, nice talk. I was just wondering, is there evidence for a role for that partial duplicate of clavata3 that you showed was maintained in the eggplant lineage?
Yeah, so you're referring to so in the eggplant we have one functional copy and most of the time a second copy that is truncated slash pseudogenized. That's the one you're
yes.
Okay. Yes. Well, so this one, this second partial duplicate is like, so used to be very likely a functional one that then underwent pseudogenization, right? So it doesn't seem that there is a function for it, at least in the maintenance of meristem, because when we inactivate the functional copy, then the phenotype is very similar to a clavata3 phenotype in tomato, by example, right? So it doesn't seem that this pseudogene or the truncated version still retains some function, but then maybe in other specific, pathways, regulation, it could be, but I think it's very unlikely.
Mm-hmm. Thanks.
Thank you.
Okay, so thank you again, Matthias. That was a fantastic talk, and really nice to see that we got a lot of audience participation this time around. That doesn't always happen, so it's great to see that people are interested. And thank you to everyone for attending. Thank you.
Thanks a lot. It was great, really. And feel free to reach out if anyone has questions or is open to collaborations. Feel free to shoot me an email. We'd be really happy to move forward.
Thanks for your time. Cheers. Bye, everyone.
Bye.